Hi, everyone, and thanks for tuning in to the Faculty of Science Undergraduate Life Sciences 3-minute thesis competition. My name is April Gislason, and I'm the research facilitator in the Faculty of Science. For today's competition, the competitors will be telling us all about the research that they've been doing this summer. The challenge for them is to showcase their research in a way that's understandable and interesting to a general audience in 3 minutes or less. So before we begin, I would like to acknowledge that the University of Manitoba campuses are located on original lands of Anishinaabe, Cree, Ojibwe, Dakota, and Dene peoples, and on the homeland of the Métis Nation. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to move forward in partnership with Indigenous communities in the spirit of reconciliation and collaboration. And I would also like to acknowledge the judges who are tuning in to the YouTube live stream, who have kindly volunteered their time to endeavor in the difficult task of choosing the winners of the 3-minute thesis competition. So there will be 2 winners that they will determine for today's competition. And the judges are Linda Alrace from the Department of Biological Sciences, Susan Egbert, who's a PhD student from the Department of Chemistry, and Dr. Hamid Riza Karbale Hideri, who is an associate professor of biochemistry and a visiting professor currently at the University of Manitoba. We also have Tiffany Penner, who is an MSc student in the Department of Microbiology, and Chloe Beret Rodriguez, who is a PhD candidate in the Department of Biological Sciences. So thank you very much for the judges for volunteering their time. And so today, the judging criteria that they will be using to determine the 3-minute thesis winners is that each competitor will be assessed on 3 criteria that are equally weighted and all have an emphasis on audience. And those are communication, comprehension, and engagement. And so with that, I will bring up our first presenter from the broadcast room here, who is Katrina Odette. And sorry, let's find you here. Hi, Katrina. Can you hear me okay?
Yeah, I can hear you.
Perfect. Okay, so Katrina has been doing research in Dr. Docker's lab in the Department of Biological Sciences, and her presentation is, Does One Size Really Fit All? Determining Ideal Filter Type and Pore Size for Use in Environmental DNA. And so I'll just bring up your slides there. Just give me a sec. Okay, so whenever you're ready.
I want you to think about all the places you've visited in the past week. How many doors and objects have you touched? Maybe you're like me and your hair gets stuck on everything, leaving a trail of DNA wherever you go. Similarly, fish leave DNA in their aquatic environment, which we call environmental DNA, or eDNA for short. It comes in many different forms, such as blood, urine, feces, eggs, and scales. eDNA is still a very new field of research, but it is becoming especially interesting from a conservation point of view. This is because it is eDNA is still a very new field of research. It is becoming especially interesting from a conservation view. This is because it is less harmful and destructive than other methods currently being used to track and identify invasive or endangered species, because it doesn't require you to physically catch the fish you are looking for. Now you must be wondering, how does eDNA actually work? Well, first you must collect a water sample using a reusable bottle. Then that water sample is run through a filter to capture the eDNA. A filter acts a lot like a spaghetti strainer to capture the it acts a lot like a spaghetti strainer that you can find in your kitchen. In this case, eDNA is like the spaghetti that remains trapped in the strainer as the water passes through the holes. Then you must remove that eDNA from the filter through the extraction process. Finally, you put the sample in a qPCR machine where it is run for an analysis. Now, my study focuses on the second step of this process. Remember, this is the one where the filter acts like a spaghetti strainer to capture the eDNA. If you're new to the eDNA field like I am, you may be overwhelmed by all of the different options you have when choosing a filter. There are many brands that exist, all with a huge range of pore sizes, from extremely small to outrageously large. So how do you go about choosing the right one? Well, my study looks at 3 different filter types and 3 different pore sizes to try and help researchers find the answer to this question. More specifically, I'm looking to make comparisons in terms of filtering time and their different abilities to capture eDNA. Also, I'm looking at different costs to see which one gives you the best bang for your buck. Finally, I believe that my study will help researchers make a more informed decision when it comes to choosing the right filter. This way, they can spend less time planning their experiments and more time doing the actual study, whether it is searching for an endangered fish or looking to see how far an invasive species has spread through our water bodies. Thank you.
Thanks, Katrina. That's super cool. Okay, so I will relieve you from the screen here. Thanks so much. And our next presenter is Jillian Wojcik. So I'll just pull her up from the room here. Hi, Jillian.
Hi.
Can you hear me okay?
I can. Can you hear me?
Yep.
Excellent.
Okay. Okay, so Jillian is doing research in Dr. McManus's lab in the Department of Biochemistry and Medical Genetics. And the title of Jillian's talk is Evaluating SCIP2 as a chromosome instability gene in colorectal cancer. So just pull up your slide. Okay, and whenever you're ready.
Did you know that in 2020, colorectal cancer was the 3rd most commonly diagnosed and 2nd most lethal cancer in Canada, accounting for nearly 12% of all cancer-related deaths among Canadians? Unfortunately, about 50% of these diagnoses occur at late stages when the 5-year survival rate can be as low as 8%. Now, despite these associations, the mutated genes driving colorectal cancer development and progression remain poorly understood. In this regard, chromosome instability is the frequent changing of chromosome numbers within our cells and is proposed to drive cancer development. While these genetic changes are associated with many cancer types, they appear in approximately 85% of colorectal cancer cases. Therefore, it is essential to characterize and identify genes that, when mutated, can cause chromosome instability, to develop new therapies, and to improve the lives and outcomes of those living with colorectal cancer. But what exactly is chromosome instability? Picture your body cells as cars, with chromosomes as the car's computer to provide the information needed to drive safely and avoid accidents. But what would happen to the car if its computer went haywire? The speedometer, the brakes, and the GPS guiding the path of the car would be critically impacted. Like cars, our cells have built-in safety features to prevent accidents from happening. But when cells experience chromosome instability, the information needed to operate normally is constantly altered due to the loss and gain of chromosomes. Cells that acquire defects in key controls like speed and GPS can behave as a runaway car and are more likely to become cancerous. The focus of my summer research has been investigating the gene SCIP2, which is frequently altered in colorectal cancer and is proposed to drive chromosome instability. Therefore, the main focus of my summer research has been to assess impact of decreased SCIP2 expression on chromosome instability in colorectal cells. Using state-of-the-art microscopes and cameras, I was able to visualize and analyze thousands of individual colorectal cells to assess hallmarks of chromosome instability, including changes in nuclear areas. The nucleus is like the software of the cell, as nuclei contain our DNA, which is able to expand and contract in order to accommodate the number of chromosomes they're holding. Therefore, large changes in nuclear areas can indicate changes in cell DNA content and therefore are suggestive of chromosome instability. To further confirm altered SCIP2 is causing chromosome instability, I was able to burst the nuclei of individual cells to count their chromosomes and compare their numbers to chromosomes in normal cells. Finally, I investigated proteins proposed to interact with SCIP2 to gain further insight into how SCIP2 regulates the mechanics of our cells. Overall, by understanding genes driving chromosome instability, we can begin to understand how to keep cells driving safely and how to correct their course when accidents happen. Thank you.
Great. Thanks so much, Sharon. Okay, so I will pull you down from this screen and we'll see you later. And our next presenter is Stephanie Chapu. So I'll just find Stephanie in the room here. Hi, Stephanie.
Hello.
How are you doing?
I'm doing well.
Good. Okay, so Stephanie has been doing her research project in Dr. Belmonte's lab in the Department of Biological Science. And her talk will be on PGP, what? The microbes feeding our future. I'll pull up your slide here. Okay. And whenever you're ready.
7.8 billion and counting. That is the number of people living on the planet right now. And like you and me, people need to eat. To meet this growing demand for food, the use of chemicals and fertilizers by farmers is a common practice, but this at the expense of harming our environment, polluting our water, and jeopardizing our own health. But to omit the use of these chemicals would mean facing economic crashes and mass starvation, which isn't any better. Luckily, thanks to modern-day research, we may have found an alternative to these chemicals, but one that is a lot smaller than you might think. Please allow me to introduce to you PGPR. That is an acronym given to a group of soil-dwelling bacteria who have been farming long before we have. These rhizobacteria have made their home in plant roots, where they harvest plant secretions to thrive. In exchange, they're able to convert chemical elements like nitrogen and phosphorus, like a living fertilizer, into forms usable by plants, promoting their growth. The thing is, there are thousands of bacteria out there, and not all of them bring the benefits PGPR do. The competition is fierce, and how do we even know what bacteria work like PGPR? And do these PGPR work with all plants? Well, that is where I come in. Over the course of the summer, my research in the lab was to investigate these questions. We would use different species of PGPR obtained from Manitoba with different crops to see how the plants, their roots in particular, were affected. Some of our experiments included growing these plants on Petri plates, as depicted on the left, where we would take seeds and put drops of PGPR species just below to see if the roots would grow faster, longer, or thicker. On the right, you can see another experiment we ran, where we would take crop seeds and coat them with the bacteria to again analyze root growth. We would do this in paper bags that allowed us to do this. Another experiment that we did, using the seed coating method, we obtained some pretty exciting results, and that is shown in the middle image. Now to the left, you see a corn that is quite smaller than the other two. This we call our negative, and it was grown in the absence of PGPR. Now notice how it's significantly smaller, and that's just with using natural bacteria, which I think is pretty crazy. So where do we go from here? Well, even after we obtain some positive results in the lab, there's still no telling how introducing these bacteria will work out there in the field in concentrated amounts. And even if they do work, a product must still be created and distributed for commercial use. There's still a lot of research left to do with these bacteria, but after my summer, and with these microbes at our side, I am positive that we are inching towards a more profitable and sustainable future in the crop industry. Thank you.
Thanks, Stephanie. That's very cool. Okay, so let's go to our next presenter, who is Rashi Chopra. So we'll find Rashi in the room here. There we go. Hi, Rashi.
Hello.
Okay, so Rashi has been doing research in Dr. Martin's lab in the Department of Psychology. And Rashi's talk is on family experience of elopement in children with autism spectrum disorder. And I'll just pull up your slide. Okay, and whenever you're ready.
Let us imagine you are out for a walk or a drive and you reach a certain point on the road that is not familiar. You don't seem to know your way back from there. What would you do? How would you feel? You would possibly try thinking of all the ways to get back home safely within time. Anxiety and stress would start to creep in. Most of us would rush for a GPS system, and some of us would even ask for directions from people around us. Now, let us imagine a child with autism spectrum disorder, or ASD for short, is in a similar or the same situation. Do you know what autism spectrum is? Any child with autism spectrum experiences a broad range of conditions, including deficits in social skills, speech, and communication. The only navigation system they have access to are their caregivers. Will they be able to identify they're no longer in a safe environment? And if so, will they be able to identify how to go back home? Unfortunately, given the deficits, no. This not only has an adverse effect on the kids, but also their parents and care providers, knowing that their child will not be able to differentiate between a safe and a dangerous environment. Elopement is defined as any dependent person leaving a safe and a supervised space of the caregiver, thereby exposing themselves to potential dangers out there. The dangers may include eloping to a water body or rushing towards the railway tracks or a busy road. According to past research, 49% of children with ASD have tried eloping, and only one-third of the families have received support from a licensed health professional. This creates a lot of distress within families. They are unable to leave home, have disrupted sleep, and a low quality of life. They struggle due to not knowing how to handle this behavior and access relevant resources. Conversely, agencies and health professionals need to understand how to offer these resources in the best possible ways, given the nature and the extent of this issue. This is where we come in. Our team is working on a project to discuss this issue of family experience of elopement in children with ASD. Our team is working to describe experience with elopement, the impact on the family, and the mitigation strategies of elopement in children with ASD. We aim to achieve these goals by conducting an online survey for families of children with ASD that are recruited by service providers. In this survey, we are asking the care providers about their experience with elopement, the impact that this has had, and also about the way they have tried to manage or cope with their child's elopement. We hope to improve the support available to the families by learning more about the aids they have or not yet received. Thank you.
Thank you, Rashi. Okay, I will pull you off the screen here and set you free. And then our next presenter, I believe, is Sharon Felix. So I'll just find Sharon here. Hi, Sharon.
Hi.
How are you doing?
Yeah, good. Can you hear me?
I can hear you very well.
Okay.
Okay, so Sharon has been doing research in Dr. Wyer's lab in the Department of Biological Science. And the title of Sharon's talk is RNAi, Our Green Savior. So I'll just pull up your slide here. And whenever you're ready.
Imagine yourself a farmer in the south of France, overlooking your beautiful vineyard, when suddenly you notice a white, fuzzy, unusual growth on your grape leaves that looks a lot like the picture on the right. You immediately sense that what you're looking at is the unfortunate attack of the plant pathogen downy mildew. Now, if you're thinking anything like the average farmer, you know a solution to getting rid of this terrible nuisance is pesticides. But pesticides have a whole list of cascading effect on not only your field, but the environment as a whole. Using pesticides also means that you can say goodbye to the biodiversity and health and well-being of your field. Another drawback to using pesticides is that they aren't a long-term solution due to the phenomenon of pesticide resistance. This is quite the dilemma. What you need is an alternative. Enter RNAi or RNA interference. Traditional fungicides target the essential proteins of pathogens, but RNAi takes a step backwards and targets the mRNA or messenger RNA that makes the protein in the first place. RNAi is a technology that reduces a gene's expression by the introduction of something called double-stranded RNA or dsRNA. Our cells and the cells of pathogens produce proteins by working through a simple chain of command, where DNA makes mRNA, which makes our protein. But what if you remove the mRNA? That's right, no mRNA means no protein. By inhibiting an intermediate of this chain, you have managed to prevent the expression of the gene, or in other words, reduce the production of the protein. Part of my research involves making this double-stranded RNA in the lab and applying it to the pathogen. The already existing machinery in the cells of the pathogen then break it down to small interfering RNA or siRNA, as you see on the left of the screen. This siRNA will then pair with its corresponding mRNA and destroy it. What's wonderful about this technology is that it does not persist in the environment like the common fungicides and pesticides used in fields today. Just imagine the vast difference this will make. We'll have healthy fields with bees, insects, and birds, minus the pathogen that we want gone. Science calls it RNAi, but I believe we might as well call it our green savior.
Thank you, Sharon. That was great. Okay, so I will pull you off the screen, and I hope you enjoy the rest of the day. And then our next presenter, I believe, is Amy. So, Amy Fernando. So let's bring up Amy.
Hi, everybody. Hi. Can you hear me okay?
I can hear you really well.
Wonderful. Okay.
Okay, so Amy's been doing research in Dr. Gordon's lab and in the Department of Human Anatomy and Cell Science. And Amy's going to talk about protecting the diabetic heart. Can roflumilast prevent cell death in cardiomyocytes exposed to lipotoxicity? And so let's bring up your slide here. Okay, whenever you're ready.
All right. Here's something really cool that you can do right now. You can take your hand and see if you can spread apart your fingers just like that. You likely can, and that's very cool because you weren't always able to do that. When you were a fetus, in the uterus, there was webbing in between your fingers. But by the time you were born, that webbing and all those cells had died. And it turns out that even as an adult, all across your body, your cells are dying. Now, most of the time, this isn't a problem. Your body knows how to handle that. But sometimes, like when someone has diabetes, that can go really wrong. And that's a big problem because Canada is in the middle of a diabetes epidemic. As many as 1 in 3 Canadians has either diabetes or pre-diabetes. So diabetes can lead to an increase in cell death, and many Canadians have it. So what? Why does it matter? Well, it turns out to be a little like real estate, in that it's the location that counts, because diabetes really likes killing the cells in your heart. And that means that cell death, a similar cell death to the one that lets you wiggle around your fingers, can actually lead to heart failure. So what can we do? That's the question I've been asking over around 2 years. We may be tempted to try to prevent diabetes, but this is really challenging. Remember that 1 in 3 of us already have or are affected by it. So what we can do instead is try to protect the heart regardless of diabetes being there. Through my work in the lab, we've identified a drug called roflumilast. Roflumilast may be the ideal candidate because it may be able to prevent those cells from ever dying. In some cell culture models of diabetes, roflumilast is able to bring down the excess cell death to insignificant levels. And this is really interesting because remember, healthy cells mean healthy hearts. And if you put that in the context of Canadians going through a diabetes epidemic, that can mean many healthier, happier, and longer lives. So if we ask the question, how can we better protect individuals with diabetes? Roflumilast may be the answer. Thank you.
Thank you, Amy. Okay, so I'll remove you from the room. Thank you so much for your presentation. And our next presenter, sorry, I've got timers and phones here. Our next presenter is Juanita Garcia. So I'll just find Juanita in the room here. Hi, Juanita.
Can you hear me well?
I can, yep. So Juanita's been doing research in Dr. Calvi's lab in the Department of Physiology. And the title of Juanita's talk is Renew Contracting. So I'll just pull up your slide here. Okay, and so whenever you're ready.
Think about the types of exercise you do. It can be walking, doing weightlifting, running, yoga, or whatever you enjoy. Tons of information exists for you to figure out how to best train to run a 10K race, lift weights, or even play rugby. But imagine you have a spinal cord injury with limited hand and trunk function and no leg movement at all. If you try to find information on how to train for a wheelchair 10K race, lift weights with no trunk muscles, or play wheelchair rugby, you would find close to none. And this is what we want to change. Exercising after spinal cord injury is essential. People with spinal cord injuries are prone to cardiovascular disease, type 2 diabetes, and obesity caused by burning many few calories a day. They are also more prone to having their leg muscles atrophy, which only brings more problems. In part, all these changes are caused by damage to the parts of the spinal cord that control our heart rate, our blood pressure, and other functions needed for exercise like sweating. Exercising goes far beyond building muscles, and it can actually help prevent many of these secondary problems that come with this injury. When we exercise, we bring our heart rate up, we sweat to cool down, and we release endorphins, which are these hormones that make us feel happier and also increase our sense of well-being. But how can someone with a spinal cord injury increase their heart rate and get these endorphins if their injury prevents them from walking and running, and they are never told what exercises they can do, for example, modified rowing, to achieve all these benefits from exercise? So for this, we chose to create a source of information to be used by people living with spinal cord injury, specifically tailored to each different level of injury. So as you can see on the slide, we have created a different picture for every spinal cord injury level to show the muscles the person can still voluntarily control, both in the upper and the lower body. The lighter color represents only a percentage of use for that muscle. For example, to flex your hand fingers, you need about 8 muscles working at the same time. For someone with a spinal cord injury, depending on their level of injury, they might not be able to have all these muscles working. So knowing which ones are the ones that they can still voluntarily control allows us to find the ways to modify the exercise and get the benefit we want. We believe that not being able to contract all of a person's muscles voluntarily due to a spinal cord injury should not completely remove the ability that we have to exercise. We also believe that there are ways to get the heart rate up, to activate pathways to get those endorphins, and get all the benefits from exercise. We hope to increase awareness and show people living with SCI that exercise will help improve functional capacity and their quality of life. Thank you.
Thanks, Juanita.
Thank you.
I will pull you down from the slide. Thanks so much for your presentation. Okay, and so our next presenter is Devin Haben. And hi, Devin.
Hi, can you hear me okay?
Yes, I can. So Devin has been doing her research project in Dr. Gerstein's lab in the departments of, Dr. Gerstein is in the departments of microbiology and statistics. And the topic that Devin's going to talk about is drug resistance and variation among yeast infection isolates. I'll just pull up your slide here. Okay, and whenever you are ready.
Did you know that most women experience at least one yeast infection in their lifetime? I bet you didn't know this fact, as women's reproductive health has been really stigmatized for a long, long time. Unfortunately, an appreciable frequency of women experience 3 or more yeast infections in a year. This can be very distressing for the female patient. But you're probably wondering, what exactly is a yeast infection? Well, normally, in the female reproductive system, there's a healthy microbiome consisting of harmless bacteria and sometimes yeast that can fend off any disease-causing bacteria or yeast from coming in. However, there can be an imbalance or a switch that causes this yeast to proliferate, which we don't understand why yet, and this therefore causes a symptomatic yeast infection. Certain treatments, such as fluconazole, which is a classic yeast infection treatment, and boric acid, which is an unconventional yeast infection treatment, are being studied and compared. Certain yeast species have become resistant to the effects of certain drugs, meaning that these drugs don't really work as well on the yeast as they used to in the past, and this can be very problematic for those seeking treatment. We can study resistance through an experiment called a disc diffusion assay, which is pictured in the photographs. We collected yeast samples from 2 different participants from the rectal and vaginal sites, therefore giving us 2 different species, Candida krusei and Candida albicans. We collect yeast from the rectum to see if it is a source for the yeast population in the vagina, but further genetic tests will have to be done. We plot the result, and each dot on the graph represents one isolate from the sample. A higher resistance is indicated on the photographs by a smaller blue line, or if the dot is higher on the graph. Results show on the left graph that there's a lot of variation in resistance between populations, but there's a smaller distribution of resistance to boric acid in comparison to fluconazole, which we're still trying to find out why. Interestingly, the graph on the right shows that in Candida albicans, resistance in vaginal samples to boric acid is slightly higher than the rectal counterparts. This is interesting because it could be indicative of a change that could be occurring in the vaginal environment, causing these yeasts to become slightly more resistant. My research aims to find new and effective treatments for women who have recurrent yeast infections. Looking at the variation in resistance has not really been done before, so this could help us find the answer as to why these women keep on having recurrent yeast infections. Hopefully, this breaks the narrative that female reproductive health is taboo and open the door to more opportunities for research to be done on female reproductive health. Thank you.
Thanks, Devin. I will pull you off the screen. Thanks so much for your talk. It's great. Okay, so our next competitor is Mohamed Hyatt. So this Hey, Mohamed.
Hey.
How are you doing?
Pretty good.
Good, good. Okay, so Mohamed's been doing research in Dr. Kress's lab in the Department of Chemistry, and the title of Mohamed's talk is Electrochemical Detection of Resistance to Tobramycin in Pseudomonas aeruginosa. And I'll bring up the slide here. Okay. Whenever you're ready.
You've just visited a doctor and they've diagnosed you with a UTI. Barring any other complications, they decide to treat you with tobramycin. Well, that's a common infection with an easy and straightforward solution. However, that answer may not always remain so straightforward. Bacteria have fought back, and some infections display resistances that make even treatable conditions seem unmanageable. For this reason, the growing problem of drug resistance has even been called the silent pandemic, a problem we've all grown a little too familiar with. For providers working with treatment plans, side effects of drugs really limit options, and drug resistances only serve to further complicate things. But imagine if we could have a sensor that could tell your doctor exactly what antibiotic was right for you. And that's exactly where my project seeks to improve, by developing a sensor that's capable of doing just that. To start, I focus primarily on the bacteria Pseudomonas aeruginosa, a species that the World Health Organization classifies as priority 1, critical for the development of new antibiotics. Now, different strains of the species display varying degrees of resistance to our antibiotic of choice, tobramycin. So what I did was incubate these different strains with our drug and then analyze the solution to see if we could even quantify that resistance. Before diving into how the bacterial species responds to the drug, we must first make sure we have a method to detect whether there is drug in solution and then accurately report its concentration. What we use to accomplish this is electrochemistry. We have a 3-electrode setup that is directly in contact with our bacterial solution. We apply an electric potential and we get a current response. More importantly, you get a resulting graph with some signals on it. You can think of the resulting graph as a sort of river. An empty or blank solution would be like observing the river flowing peacefully. However, once the potential is applied, and if there is a drug present, then the river will behave a little bit more chaotically. We would see additional eddies and currents that show up as peaks on our graph. On top of that, these peaks are proportional to the amount of drug in solution. Now, the bacteria themselves can respond to tobramycin in 2 ways. If a strain expels it, we would see an increase in current response or a larger peak. But if the bacteria uptakes the drug, then we would see a decrease in current response. If our solutions end up displaying different tobramycin concentrations, this could indicate that one strain may be uptaking less antibiotic than the other, a direct result of resistance. This would be a desirable result, validating that our sensor can tell us how resistant a strain is. Future experimentation could then focus on determining this in live samples, with the goal of an improved outlook for treating infections from Pseudomonas aeruginosa, providing one small step towards surpassing bacteria in the fight against drug resistance. Thank you.
Thank you, Mohamed. Okay, so I will pull you down. Thanks so much for your presentation. And our next presenter is Kusha Kamal. So I'll just pull up Kusha. Hi, Kusha.
Hello.
How are you?
Doing great.
Good, good. Okay, so Kusha is doing research in Dr. Wired's lab in the Department of Biological Sciences. And the title of Kusha's talk is A Species-Specific Approach to Mosquito Population Control, which is aptly timed. And there's your slides. So just give me 1 sec, and whenever you're ready.
Imagine you're walking down the streets on a beautiful sunny afternoon when you look up at the sky and notice a fleet of drones releasing what looks to be a massive swarm of mosquitoes into the air. What if I told you that this isn't the plot of an upcoming horror movie, but it's actually a very safe and effective way of controlling mosquito populations in the region? It's called the sterile insect technique, and it involves releasing millions of non-biting sterilized male mosquitoes into an area where they could mate with the native females of a population, thereby reducing the number of viable progeny produced in each generation. You see, female mosquitoes bite humans, and as a result, are one of the most prominent disease vectors in the world, infecting people with deadly pathogens that kill nearly 1 million people every year. In the absence of effective vaccines for many of the illnesses that they lead to, like malaria, Zika, and dengue, our main response against their spread involves the use of broad-sweeping insecticides. These chemicals are known to negatively impact non-target insect species that we care about and can also lead to some mosquito populations becoming resistant to their effects over time. On the other hand, the sterile insect technique is an environmentally friendly and species-specific approach that has shown to be very effective in eradicating several different pest species from various regions of the world. However, it has never been as successful when it comes to mosquitoes, mainly because of the drawbacks associated with conventional sterilization methods, like the use of chemical sterilants or X-ray radiation on the mating performance of these males when they're released. So to overcome this limitation, my work with the Wired Lab involves looking into RNA interference, which is a genetics tool that allows us to reduce the expression of specific genes in an organism by introducing double-stranded RNA molecules into their system. In this case, we can actually feed our mosquito larvae a combination of double-stranded RNAs that target both female development and male fertility at the same time, allowing us to simultaneously sex sort and sterilize our male mosquitoes in the lab without the use of any dangerous chemicals or expensive X-ray machinery. This approach has the potential to significantly cut down on costs associated with mass rearing sterile males, which in turn could bring this technique one step closer to being implemented on a larger scale and becoming commonplace in the near future. Thank you so much for listening. I'll hand it off back to April.
Thanks so much, Kusha. Okay, so I'll pull you down at the screen here. And so our next presenter is Matthew Kirby. Hi, Matthew.
Hi, April.
How are you doing?
Pretty good. Yourself?
Good, good. Okay, so Matthew has been working in Dr. Stepfeld's lab in the Department of Chemistry. And Matthew's project is signal jamming the computational design of Netrin-1 DCC inhibitors. I apologize for the phone ringing in the background. I'll just pull up your slide here. Okay, and whenever you're ready.
I'd like to introduce you to Netrin-1, a protein involved in all manner of processes, from cellular movement to neurological development, but most notably, Netrin-1 plays a prominent role in many cancers. Being upregulated, that means overproduced, in over 80% of all known human cancers. Now, to understand why that is, you should know that your cells that make up your body are very needy. They need constant encouragement to survive, to grow, to function. Netrin-1 is that encouraging signal. It's like a cellular pat on the back, so to say. So when it binds its receptor, in this case DCC, the cell survives, it goes about its day, everything's good. But when Netrin-1 is absent and DCC is unbound, your cells default to performing a programmed cell death for fear that they might have picked up a mutation, a genetic defect, that could render them potentially cancerous. So if you're a cancer cell, like all biological things, you want to survive for as long as possible and to replicate as much as possible. So one of the best ways to do that is to overproduce a survival signal like Netrin-1. So now that we know that, and that brings me to my research. We have, at the Settlefield Loud, a general dislike of cancer. So the next step after we figured out this interface was to ask, can we block this Netrin-DCC interaction from occurring? Can we trick cancer cells into killing themselves thinking that Netrin-1 isn't present? But how do you do that? Well, the answer apparently involves a combination of computers, proteins, and chemistry. Now that might seem like an odd combination at first, but you have to consider that computers are really, really good at performing repetitive calculations quickly and correctly. And us as humans have gotten pretty good at describing our world in numbers. So starting from a single protein picture, we can ask a program to map where a potential drug might bind. We can also ask a software to put a drug onto our protein and ask it, how well does it bind? How specific is it? We can ask a dynamic simulation to show us, how does it block? How does it interact? Does this drug actually work? And you can do that hundreds of thousands of times over and over and over again while, you're in the lab. You bring that stuff out and you optimize it, and that'll save you years of work. What used to take days, weeks, months, years, now takes seconds, minutes, and hours. That process, that's time-saving process, that could mean a huge difference, especially when you're dealing with something like cancer that could save millions and millions of lives. Thank you very much.
Thank you, Matthew.
Thanks.
So we'll take you back to the screen here. Thanks so much. And our next presenter is Patrick Lloyd. So let's find Patrick in the room here. We're just over the halfway mark for our competitors. Hi,
Hello?
Patrick. Can you hear me okay?
Yep, can you hear me?
Okay, so Patrick is doing a research project in Dr. Belmonte's lab in the Department of Biological Sciences. And the title of Patrick's talk is Synthetic Fertilizers, Who Needs Them? So I'll just pull up your slide here. Okay, and just whenever you're ready.
Have you ever been camping, canoeing, hiking on a warm, sunny day, and all you want to do is get to the nearest lake or beach and jump in the water, but you get there and you find this algae advisory sign? Well, nitrogen and phosphorus pollution is a pressing problem for the ecosystem health and climate. Large amounts of nitrogen and phosphorus are applied to farms as synthetic fertilizers, which can get washed into bodies of water, causing algae blooms, which are toxic, killing off marine life, and they contribute to greenhouse gas emissions. Continued use of fertilizers alter soil composition over time, like hardening the soil, destroying microbial communities, and can cause high salt and acidity levels, which is a bad growing environment for future crops. So, after hearing this, wouldn't you want to reduce our dependencies on these synthetics? The solution could come from your own backyard. You just need to do a little bit of digging, but don't worry, we've already done that. In the lab, we are working with bacteria that have been isolated right here from Manitoba soils and utilizing them as plant growth promoters, eliminating or reducing the need for synthetic fertilizers. We are working on a bacterial solution to increase plant growth, where we essentially cover the seed with the solution, allowing for a symbiotic relationship to form, which is beneficial to the plant and bacteria. After growth, we then check biometrics, which essentially means we weigh and measure the roots and shoots to see if the solution increased growth or not, or promoted growth or not. Also, bacteria replenish the soil by breaking down organic matter and cycling nutrients over time, which fertilizers cannot do. Utilizing bacteria as a plant growth promoter eliminates the fear of any negative downstream effects because the bacteria is already an inhabitant of the soil. So, to keep people, wildlife, lakes, and beaches safe, we need to find on better ways to protect them. This means using new and innovative ways, like using our local Manitoba bacteria, which will improve crop development, improve crop yield, and improve the farmer's bottom line. So, synthetic fertilizers, who needs them?
Thanks, Patrick. That's great. Okay, so I will take you off the screen here. Thanks so much for your presentation. And so our next presenter is Elise Malasso. And
Hi.
hi, Elise.
Yeah, can you hear me well?
I can, yep.
Okay, awesome.
So Elise has been working in Dr. Sharif's lab in the Department of Pathology. And the title of Elise's talk is Exploring the Role of Metabolism in Glioblastoma Relapse. Let's go through slide. Okay, and whenever you're ready.
Glioblastoma is a very devastating form of brain cancer with one of the worst survival rates of any cancer. It is very deadly, with less than 5% of glioblastoma patients in Canada surviving within 5 years. What makes this worse is that it is the most common brain tumor diagnosed in adults, and it is highly invasive, spreading throughout the brain rapidly. So, surgery is done to remove the tumor, followed by aggressive radiation and chemotherapy. But sadly, for these patients, cancer relapse is almost always a certainty. Now, why is this? A major problem is that glioblastoma tumors have a mixed population of cells that respond differently to therapies. Cancer stem cells are cells that are more resistant to chemotherapy and can keep making more copies of themselves indefinitely, causing tumor relapse. Our lab aims to discover a combination of therapeutic strategies to treat glioblastoma patients more safely and more effectively to minimize toxic side effects. What makes our research special is that we are using brain tumor cells from glioblastoma patients that had their first tumor surgically removed after diagnosis and had their tumor removed after they suffered from a relapse. So this is our primary and recurrent tumor samples, making our research findings more clinically relevant. Ultimately, we are focused on understanding how glioblastoma cells adapt to chemoradiotherapy by changing their metabolism or their energy production to promote tumor relapse. With matched primary and recurrent tumors, we are looking into what metabolic changes occur going from a primary to a recurrent tumor. We also created a resistant cell line by treating glioblastoma cells with the same radiation and chemotherapy used in clinics to treat patients. Interestingly, we discovered that in these cells, they adapt by rewiring energy production to rely less on glycolysis, which is the main energy production and cell pathways of cells, and instead to rely more heavily on mitochondrial production, another energy production pathway. Targeting increased mitochondrial production may make these glioblastoma cells more responsive to chemoradiotherapy, and it may serve as an ideal target to overcome glioblastoma chemoresistance. As standard treatments have poor survival outcomes, it is very, very important to identify new therapies to increase patients' chance, to increase their chance of long-term survival and to decrease their occurrence of cancer relapse. Thank you.
Thanks, Elise. Okay, so I will take you off the screen here. Thanks again for your presentation. Okay, so our next presenter is Japji Matharu. So, hi, Japji.
Can you hear me?
I can, yep. So Japji has been working in Dr. Wired's lab in the Department of Biological Sciences. And the title of Japji's talk is Accessing the Stability of Double-Stranded RNA in Egypti to develop a species-specific pesticide. So I'll pull up your slide here. And whenever you're ready.
Imagine returning home after a refreshing, much-needed vacation to Cuba when suddenly you are hit with an intense headache. Over the next few weeks, your symptoms spiral out of control. You are rushed to the hospital after suffering from a seizure, and doctors tell your family that you are experiencing multiple organ failure and you may not make it. They say that you have dengue fever, which you contracted from a mosquito bite during your trip. This scenario is the reality for thousands of people each year. The Aedes aegypti mosquito is the deadliest animal on the planet, being responsible for the deaths of hundreds of thousands of people each year. It is a vector for diseases such as dengue, West Nile virus, yellow fever, and Zika virus. A vector is a vehicle that carries and transmits diseases from one organism to the next. The Aedes aegypti is a major threat for those who live in tropical areas. That is 40% of the world's population. In addition to being largely ineffective, current pesticides are very harmful to the environment. Outbreaks still occur, and many off-target species are killed. We must revolutionize the technique by which we control the population of this deadly mosquito. In my lab, we are aiming to create an environmentally friendly and species-specific pesticide using RNA interference technology. RNA interference is a mechanism that inhibits gene expression at the translational level. Specific messenger RNAs are targeted and destroyed, thereby preventing gene translation into protein. As a result, a gene is effectively silenced. My project involves assessing the stability of different double-stranded RNA molecules in the guts of the Aedes aegypti. I have been collecting larvae guts, as seen in the left part of my slide, and I've been adding them to different double-stranded RNA molecules and assessing the degradation of each molecule over time. My experiments have proven to me that the paperclip form is the most stable, as depicted in the graph on my slide, and therefore it should be used to develop a pesticide by using in vitro transcription. In vitro transcription is the process of administering double-stranded RNA to an organism externally, such as by feeding. The pesticide that will be developed will be organic, and it won't have any off-target effects because the double-stranded RNA is targeted specifically for genes within the Aedes aegypti. Preventing deaths as a result of a simple mosquito bite should not come at the expense of the environment. RNA interference has the potential to overcome this deficiency. Thank you.
Thank you, Japji. Okay. I will pull you down. Thanks so much for your presentation. And our next presenter is Talani So I'll just put Talani. Okay.
Hi, April.
Hi, Talani. So Talani Ola Rawaju. I'm trying to say the name properly. I apologize. So Talani has been working with Dr. Coombs from the Department of Microbiology, and the title of Talani's talk is Photodynamic Inactivation Effect on Influenza using TLD 1433 as a photosensitizer. So I'll just pull up your slide. Okay, and whenever you're ready.
Okay, picture this. Person A gets a COVID-19 vaccine on a Tuesday. She then got notified about a potential contact with a positive COVID case on a Thursday, which is also the day she got tested. By the Friday, she gets a positive result back. Person A was me. And although I had gotten the first shot of the COVID-19 vaccine days prior to getting tested, I still experienced symptoms of COVID-19. This example shows the difference between vaccines and antivirals. Vaccines help provide protection before you get infected, while antivirals can help reduce viral load and also serve to prevent spread of the infection to others after you've gotten infected. There are currently no approved antivirals for COVID-19, while there are just only a few for the flu. This is where my project comes in. My project aims to study the effect of a photoactive compound on influenza virus, which is the virus that causes flu, using a treatment called photodynamic therapy. As pictured on the screen, in photodynamic therapy, light is shone on a photoactive compound, also called a photosensitizer, which then absorbs light at a specific wavelength and gets excited. This excited photosensitizer can then react with molecular oxygen, creating reactive oxygen species, which are oxygen compounds that are highly reactive. This reactive oxygen species can then react with biomolecules in organisms, including carbohydrate, proteins, lipids, and nucleic acids, and causing destruction of these organisms, in my project case of the influenza virus. My lab cannot work directly on SARS-CoV-2, which is the virus that causes COVID-19, because we are not a biosafety level 3 lab. However, influenza virus, like SARS-CoV-2, is an envelope virus, which means its genetic material is wrapped in an envelope. Previous studies done studying the effect of this photoactive compound as a disinfectant on model viruses, including influenza virus, Zika virus, and a coronavirus strain, shows that this compound is effective in the nanomolar range, which is nothing like has been seen for most disinfectants previously. The results of my project will be able to determine potential antiviral and treatment for COVID-19 and flu, and hopefully help with people with severe cases of these infections and create a better quality of life. Thank you.
Thank you, Talani. Okay. So I will take you off the screen. Thanks again to Talani. And our next presenter is Zeal Patel. I think Zeal might be having some technical issues. Let's give him a minute. There we go.
Hi.
Hi, Zeal.
Hi, how are you doing?
Good. Can you hear me okay?
Yeah.
Okay. So Zeal has been working in Dr. Resnick's lab in the Department of Microbiology. And the title of Zeal's talk is Overcoming Nitrogen Pollution Through Rhizobium Lagoon Symbiosis. I'll just pull up your slide here. Okay, and whenever you're ready.
At the end of 2021, the world population will reach 7.9 billion. And at the end of 2030, the world population will reach 8.5 billion. While in 2050, we are projected to reach around 10 billion. So, how to feed 10 billion people is going to be the biggest challenge. And the bigger question is how to feed 10 billion people in a sustainable manner. By the word sustainable, I mean pollution-free as well as cost-effective. So to give you in perspective, 41% of the water pollution in Gulf of Mexico is caused by fertilizers. And nitrogen is the major compound of fertilizers. Due to this, there is a huge deposits of nitrogen in the water bodies, which promote the growth of algae and aquatic plants. These algae and aquatic plants compete with aquatic animals in terms of dissolved oxygen and nutrition, which leads to the decline in the population of aquatic animals, and hence eutrophication. And as we all know, eutrophication is the major factor of water pollution. So how to control this? So thankfully, we have two options. First option, to reduce the use of fertilizers. But since we have to feed 10 billion people till 2050, that's not an option. So second option is working with the legume plants and with the keen interest in rhizobium legume symbiosis. Why legume plants? It's a general question, right? Again, two reasons. First reason, their natural ability to fix nitrogen. With the help of rhizobia, they can fix nitrogen. And as we know, rhizobia are the gram-negative bacteria which colonizes the root cell of legume plants to fix nitrogen. And in return, they get carbon source from the plant. It's a similar relationship of bee and flower, where bee gets the nectar and flower gets to disperse their pollen grain. And the second reason is, when we isolated the bacteria from the soil of legume plant, we found out that there is a consistency in the microorganisms from different locations. It means that there is some relationship with the microbial diversity and the plant growth. So if we can exploit those microorganisms in such a way that we can enhance the nitrogen fixing capability of rhizobia and manipulate the microorganism in the soil in such a way that it promotes the plant growth, then we won't need fertilizers. And that will be equivalent to driving a car without ever refueling it. Not just that, we can also apply our knowledge and understanding for non-leguminous plant. Thank you.
Thank you. Okay. I will pull you down and set you free for the rest of the day. Thanks again. Okay, and our next presenter is Randy Roy. And so Randy, just pull you up here. Hi, Randy.
How's it going?
Good, how are you?
How is the volume?
The volume is good now, yeah. Yeah, perfect. Okay, so Randy's been doing research in Dr. Sorensen's lab in the Department of Chemistry. And the title of Randy's talk is Unlocking the Potential of the Fungal Secondary Metabolome. There's your slide. So whenever you're ready.
Did you know that over 90% of the antibiotics used today come from natural products? You may be wondering, what is a natural product? Natural products are small organic molecules that come from microorganisms. One of the largest studied microorganisms is fungi. The most well-known example of an antibiotic produced by a fungus is the discovery of penicillin by Alexander Fleming. Penicillin directly changed the path of medicine, and not just by treating some bacterial infections, but it inspired the discovery of hundreds of other antibiotics. However, over the years, there has been an increasing need to discover new antibiotics due to new emerging diseases and antibiotic resistance. So where do we go from here? The fungal natural product metabolome is a rich reservoir for new antibiotics. It has been reported that there are over 1.5 million species of fungi, and only 10% are known within the scientific community. And within that 10%, only a small fraction have had their natural products characterized. With so much left to uncover, how can we unlock this potential? By using traditional techniques, we are able to unlock and discover new natural products. My story starts with a fungus that lived within a unique ecological niche in the soil from White Shell Provincial Park under a lichen. We wanted to see what natural products this fungus would produce. When cultivating in the lab under optimal growth conditions, we noticed a unique characteristic. This fungus was able to excrete a yellow pigment into its environment, whether that be on solid or liquid media. I was able to isolate and purify this compound as well. Using DNA sequencing, I was able to identify the name of the fungus, Trichoderma citronviridae. My yellow molecule has shown antimicrobial activity against pathogenic strains of bacteria and shows promise for antifungal activity as well, but further assays are required. Using spectroscopic techniques, by the end of the summer, I will be able to elucidate the chemical structure of this molecule. With over 90% of fungus unknown to the scientific community, there is a huge potential for new antibiotics that needs to be explored. With my yellow molecule showing antimicrobial activity, I'm taking a small step in the search for the next penicillin. Thank you.
Thanks, Randy. That's great. Okay, I will take you off the screen. Thanks again for your presentation. Okay, and our next presenter is Megan Smith. And so I'll just have Megan here. Hi, Megan.
Hi, how are you?
I'm great. How are you?
Good.
Good, good. Okay, so Megan has been working in Dr. Bacher's lab in the Department of Microbiology this summer. And the title of Megan's talk is Managing Mycotoxins, Understanding Nutrient Acquisition in Fusarium graminearum. And I'll just pull up your slide here. Okay, and whenever you're ready.
I want you to think about what you've eaten in the past few days. The weather has been great, so you might have had a cold beer with some friends, or maybe you made a sandwich to bring for lunch. Like most of us, you've likely enjoyed some cereal grain products recently, like barley and wheat. But what most of us don't know is that behind the doors of your favorite brewing company, there is a serious fight against a fungus, and its name is Fusarium graminearum. Fusarium is responsible for Fusarium head blight, a devastating disease that decreases both grain yield and quality and ultimately results in massive economic losses of billions of dollars for the food and agriculture industries. But if I haven't convinced you of the severity of Fusarium infections yet, we need to discuss mycotoxins. These are toxic chemicals produced by Fusarium that can contaminate the food supply of both animals and humans. But the consequences of ingesting these mycotoxins include nausea, diarrhea, fever, and reproductive health issues, just to name a few. To make matters even more complicated, the mycotoxins produced by Fusarium are extremely resilient to any attempt to eliminate them. So it's important to minimize the growth of the fungus in order to manage the mycotoxins. And that is where the research in my lab comes into play. My project focuses on understanding how Fusarium obtains its phosphorus supply. But why phosphorus, you may be wondering? Well, phosphorus is an element that is absolutely essential to Fusarium survival. So, if we can learn how it succeeds, we can take that information and force it to fail. When the fungus cannot get its phosphorus, its growth will be inhibited, and we can decrease any dangerous mycotoxins that are lingering in our grain. Now, I want you to think about imagining a group of people playing Jenga. You can identify the block that nobody wants to remove because it stabilizes the entire tower, and once it's removed, the tower crumbles. The fungus's ability to supply itself with phosphorus is that special block that it doesn't want anyone to touch, because that is the block that lets it thrive on our grain. But I want to ruin its game. I want to understand how that piece works, and eventually, I'd like to remove it. With this information, I hope to contribute a critical piece of knowledge to a larger effort of managing Fusarium. This includes the development of different management strategies, such as fungicides, biocontrol agents, and strategic cropping strategies. If these strategies are refined, we can make the tower fall and force the fungus to fail. This will ultimately contribute to higher quality grain that is safe for consumption, so you don't have to worry about any toxins in your toast. Thank you.
Thanks, Meg. Okay. Thank you so much for that presentation. I will pull you off the screen now. And our next and second last competitor is Shabneed. And just find Shabneed here in the room.
Hi, can you hear me okay?
I can hear you very well, yeah.
Okay.
So Shabneed has been working in Dr. Kusha's lab in the Department of Chemistry this summer. And the title of Shabneed's talk is Cytochrome C Oxidase Deficiency Detection Using Scanning Electrochemical Microscopy in Living Cells. And I'll just pull up your slide here. Okay, and whenever you're ready.
Okay. A 4-week-old baby boy is admitted to the hospital because of severe muscle weakness. At 11 weeks, he struggles to breathe independently. At 14 weeks, he passes away. This is the sad reality for many babies and infants living with cytochrome C oxidase deficiency, or COX deficiency for short. In fact, 2/3 of infants die within months of diagnosis. Cytochrome C oxidase deficiency is an inherited disorder characterized by the absence or abnormality of the protein cytochrome C oxidase, resulting in severe muscle weakness, heart, liver, and kidney problems, and brain damage. Cytochrome C oxidase is a protein that is found in the membrane of the mitochondria of the living cell. It is an essential part of the energy-producing process. And to make matters worse, there's no cure for this disorder. However, the resulting symptoms can be managed and improved upon if early diagnosis is made. And this is a problem. Currently, this disorder is being diagnosed through clinical tests and through muscle biopsies, which are painful, invasive, expensive, and time-consuming, leading to a delay in diagnosis. Just like how a diabetic patient can use a glucometer or a glucose biosensor to track their blood glucose levels, what if the diagnosis of COX deficiency was seconds away? What if within seconds you could tell if a child was cytochrome C oxidase deficient or not? And this is the aim of my research. Using electrochemical methods, specifically scanning electrochemical microscopy, or SECM, we can detect any electron transfers occurring during oxidation and reduction. We're using a compound called TMPD because it's known to indicate whether bacteria can have cytochrome C oxidase or not. And then we want to apply this to living cells. By taking our microelectrode, we can scan over the cells that are in the TMPD solution. So the cytochrome C oxidase oxidizes the TMPD, and we reduce the TMPD back to TMPD plus back to TMPD. So we are able to see that this is a loop that can be detected as a change in current, allowing us to observe the cytochrome C oxidase activity. In fact, this is what our current experiments show, and this can be seen in the graph on the screen. In short, the development of a biosensor using electrochemical methods like SECM becomes a highly effective, cost-efficient, and quick means of detection for cytochrome C oxidase deficiency, allowing for early intervention, which can save the lives of thousands of children. Thank you.
Thanks, Shabneed. Okay. I will pull you off the screen. Thanks so much for your presentation. And our final presenter for the day is Milac Yossi. Hi, Milac. Sorry, I think you're muted, but can you hear me?
Yeah, I can hear you.
Okay, good. I can hear you as well. Perfect. So Milac is also in Dr. Kusha's lab in the Department of Chemistry, and will be presenting about the electrochemical characterization of the auranopin in aqueous media.
Yeah.
Uranopin, I think, was how I'm supposed to say it, sorry. And here's your slide, and whenever you're ready.
In 1928, Alexander Fleming discovered the first antibiotic, penicillin, in what is described as the single greatest victory against disease. Antibiotics brought medicine to the modern age, treating previously deadly infections easily. But overuse of antibiotics has a selective pressure on bacteria, making them stronger. That leads to the global issue of antibiotic resistance, where we are creating stronger and stronger drugs to fight ever stronger and more resistant bacteria. In the very near future, we might go to the dark ages where a simple finger cut may be lethal and surgery is far riskier because of possible infections. Currently, 10 million people die every year due to drug resistance worldwide. The WHO projects in just the next decade this number could double. Fast detection of antibiotics in sewage, rivers, human blood, etc., help curb the development of bacteria resistance. In recent years, electrochemistry has emerged as a reliable and speedy method of detection. It offers nanomolar level sensitivity and is able to detect trace amounts of an antibiotic, also being able to quantify drug resistance in a particular bacteria strain. This summer, I identified the antibiotic uranofin using a carbon sensor. Uranofin is a gold-based antibiotic effective in inhibiting cancers such as leukemia and ovarian, and effective against various parasites that have grown resistant to other treatments. It's also effective in lowering the transmission level of viruses such as HIV and COVID. Uranofin also boosts other antibiotics, making them more effective and to fight the superbacteria. I've visualized the identification of uranofin in the bottom left image. As you can see, a potential is applied to solution of uranofin by a carbon biosensor. This potential will reduce, giving electrons to the uranofin molecule, and then oxidize, taking the electrons away from uranofin. The detection level is related to the concentration of uranofin molecules in that solution, meaning if there's more uranofin, there's more electron transfers happening in the surface of the electrode, and then we're given a higher peak in the two graphs, as you can see over here on the bottom right concentration graph. The carbon-coated biosensor can differentiate between a blank solution and 200 nanomolars of uranofin, meaning if you dropped a gram of uranofin in 7,000 liters of water, that sensor will be able to detect it. This sensor works very well in acidic, neutral, and alkaline environments, making it useful for detection in a variety of environments. The sensor is also very cheap and portable, making it perfect for developing countries which are most impacted by the antibiotic resistance. Thank you for coming to my talk.
Thanks, Milac.
Yeah.
Okay, so I will take you off the screen, and you are our last presenter of the day. Thank you very much for your presentation. Okay, so that's it for today's 3-minute thesis competition. Tomorrow we'll be holding the physical science, computational, mathematical, and statistical sciences competitions at 1 o'clock. And it will be live streamed through YouTube again. I wanted to say thank you so much for your attention, everyone that tuned in live to watch these competitors. Special thank you to the judges for volunteering your time, and I will be meeting with you shortly for you to deliberate the winners. And most importantly, thank you to all of the students who presented their very cool research. You did a fantastic job, and you should be proud of yourself. So, and also just a quick reminder to the students to go on Learn to vote for your favorite 3-minute thesis presentation for the Students' Choice Award. Okay, thanks everyone. Have a great afternoon.